All right, good morning and good afternoon to everybody. My name is Blake Anson. I am at Cellular Dynamics, and I welcome you to our webinar, where we will discuss rapid and reliable neurotoxicity detection with myself and Dr. Benjamin Bader from Neuroproof, GmbH. We're going to really focus in today on looking at how stem cell-derived neurons can provide multiple answers to various forms of neurotoxicity. And we'll be focusing here on both GABAneurons as well as GlutaNeurons. All right, questions are welcome. And in order to ask a question, you'll log it in, and then we'll address as many as we can at the end of the seminar. So in order to answer or ask a question, first of all, you want to open the dialog box and click on the arrow such that it points to the right, and you'll see a little drop-down menu. At which point, go into the question arrowhead, press that so it points down, and you'll enter, or you'll see a spot for your question to be entered. All right, we then enter the question, press enter, and at the end of the webinar, we can go ahead and then answer them. You can also submit questions to either Ben at Neuroproof or CDI at support@cellulardynamics.com, and those email addresses are down here on the lower left-hand side of the screen. All right, and so with that, I think we'll just kick it off. What's going to happen here is I'm going to give an overview, a little bit of CDI, and of two of our neuronal products, GABAneurons and GlutaNeurons. Dr. Bader will then go in and talk in detail about the high-level and complex analysis algorithms they've used or they've developed to detect seizurogenic activity, as well as how to classify various types of neurons and their responses to compounds. So we're really looking at the first part of the webinar is talking about a reagent. The second part of the webinar will be talking about how you actually implement that reagent in toxicity testing. All right, so moving on, probably everybody here understands the power of iPSC technology. That is where you take the human sample, send it through a series of laboratory manipulations to make stem cells, and then you can differentiate those stem cells into a variety of terminally differentiated cells for use in basic research, toxicity testing, drug discovery, and cell therapy. And the real advantage here then is being able to translate that data back to the human condition, because again, that is your starting material. At CDI, we're a leader in the stem cell technology, and we are split into two different divisions: a life science tools division, which we'll be talking about today, as well as a cellular therapeutics research division. Our core competencies are the reprogramming of the iPSCs and genetic engineering of those lines, from the scalable manufacture and differentiation to various cell types, as well as then phenotypic assay development. And from an assay development standpoint, we develop the assays with the intent to transfer them to a third party. We also have an extensive IP portfolio, and we are a wholly owned subsidiary of Fujifilm with worldwide footprint and distributions. All right, within our life science division, we break that up into three different areas. One is the iCell products. These are our standard off-the-shelf tissue-specific cell types. So whether they're from neuro, hepato, or cardiac lineages or others, these are the standard off-the-shelf offerings. We also have what are MyCell products and custom manufacturing products. And this really takes advantage of the power of iPSC, where you can go to targeted populations and generate that research material. So whether it's looking at differences among different genders or populations, clinical populations, or disease types, you're able to make that translation and make that leap and use that broad base of genetic material, or sorry, cellular material from actual relevant populations. At CDI, we currently offer 14 different iCell types, and these are across neuronal, hepatic, cardiac, and blood lineages. All right, all of them are made to a stringent set of quality control standards, and we have a fairly large compendium of validated uses within the literature, well over 100 to 150 examples of peer-reviewed papers. All right, we're going to focus in on the neuroscience portfolio, and at CDI that consists of GABAneurons, DopaNeurons, astrocytes, and on the next page we'll see a couple more. But the point here is that all of these different cell types are provided with a variety of gene expression, marker expression, identifying characteristics, as well as varying levels of functional analyses. Right? As I said, to round out the neuronal portfolio, we've got the GlutaNeurons, Glutamatergic neurons, and motor neurons. So again, very cell type specific neuronal lineages. All right, the power of the iPSCs, as you can see, maybe somewhat choppy with this movie, is the recapitulation of that relevant cellular behavior. So in the case of neurons, what we see is the neurons settle down, adhere to the bottom of the dish, and then branch out, spread out their processes, seeking out the correct partners, partnering with those other cell types, and as we'll see later, beginning to communicate with those other cell types, right? And what this allows us is to recreate the biology, but more importantly, to interrogate that biology. And that's something that Dr. Bader will really go into in quite a bit of detail. All right, we have a wide variety of applications across the iCell products, across the neural products. That includes ways to modify the cells and ways to assess the cell health, as well as their function, and ultimately how those three areas can be used in toxicity and drug development. We're going to be focusing on toxicity here, and I'm going to focus in on GABAergic neurons as well as glutamatergic neurons. So the GABAneurons, as the name implies, are primarily GABAergic, and we can see them being used in a variety of neurodegenerative diseases, safety pharmacology, as well as compound screening and drug development. I'm going to focus in on a couple of examples from the literature here of safety pharmacology and neurotoxicity. First one really takes advantage of the structural, being able to interrogate the structure of the cells and look at structural toxicity, specifically here looking at neurite outgrowth. This is work that's come out of the Sanford Burnham and Bang's lab, where they use high content imaging to look at, assess, and measure and quantitate neuronal outgrowth and modification of neurites in response to compounds. So it's fairly straightforward, where you acquire an image, you detect the various aspects of the neurons, and then you pull out a variety of features, including nuclear features, as well as neurite features. The point is, you're able to quantitate these previously qualitative aspects. Work that's come out of her lab and was presented a few years ago at ISSCR looks at a screen of 5,200 compounds to look at changes in neurite outgrowth and morphology. And the power here is that they've identified novel hits, both for outgrowth as well as for retraction or toxicity. Right? So we're really bringing this unique biology into the dish and into interrogatable fashions. Okay. Investigators at the National Toxicology Program in the United States went ahead and ended up multiplexing additional assays on top of this neurite outgrowth. So here they looked at neurite outgrowth, viability, as well as mitochondrial effects, as a way for novel assessment of developmental neurotoxicity. And they're able to rank order compounds as well as look at specific mechanisms of action. So whether the cells are just dying or whether they're showing specific types of toxicities across the different modalities. Again, it's a way of holistic interrogation to pull out the multiple mechanisms of toxicity. All right. Eileen Dolan at the University of Chicago has taken this technique of analysis and really applied it to chemotherapy-induced peripheral neuropathy. This is an adverse outcome that affects clinic, that is seen in the clinic in a subset of patients that severely limits their therapeutic options. So it's a very crucial not disease endpoint, but therapeutic endpoint that one wants to avoid. What she showed is that the iCell GABANeurons are very sensitive to a variety of the chemotherapeutic agents. Via high content imaging, she was able to quantify and pull out multiple aspects of the neurite morphology. But more importantly, as we can see down here in panel H, which is looking at cell body area, these effects are not generalized cell death. They really have to do with the neurite outgrowth. So again, they've got, in theory, the relevance to the clinic. She went on and demonstrated that relevance to the clinic by doing another set of studies where she knocked down the function of a gene that, when knocked down, is known to impart sensitivity to chemotherapy, chemotherapy agents. So the gene CEP72, people that have an inherited polymorphism in that gene are more sensitive to chemotherapeutic agents. And she showed that in iCell neurons, when you knock down that gene through siRNA, you see an increased sensitivity. So not only do we see an in vitro phenomenon here, we also see that translation to the clinic due to the human nature of the cells. All right, so to recapitulate or to review on the GABAneurons, we can see that we've got that normal neuronal biology, the demonstrated model for neurite outgrowth with clinical translation, and therefore it really provides an unattainable model, previously unattainable model for studying human neuronal neuropathies, developmental biology, and structural toxicities. All right, that part all dealt with the structure of the neurons. Now we're going to look at electrical excitability. And again, I'll give a brief overview, and Dr. Bader will actually take a deeper dive into how one can do the analysis of really a complex biological phenomenon. All right, so we're moving into the glutamatergic neurons, or iCell GlutaNeurons. Again, we see a wide variety of uses for them. We're going to be focusing in on safety farm and seizurogenic models. As this is a newer product, I'd like to spend a couple of slides on the characterization. So under the light microscope and with fluorescent labeling, you see a very typical neuronal morphology where you've got the soma and a multipolar outgrowth of neurites. And this occurs within days or even hours of plating. It's possible to make many different lineages, neuronal lineages. So we want to ensure that we are looking at the cells we think we're looking at. So we do that through a variety of marker analyses. First is to demonstrate the neuronal identity through TUJ and MAP2 staining. And then secondly is to verify the glutamatergic identity. And this we do through single cell expression analysis via the fluidigm. What we're looking at here is a heat map of the expression. We've got around 50 cells on the x-axis. On the y-axis are all the various genes. And we can see up here on top, we've got the glutamatergic markers, and down below, we've got GABAergic markers. And we can see we're sitting at about 75 to 80% glutamatergic neurons. So we've got a highly pure neuronal population that is greater than 70% glutamatergic in identity. Right? Now the whole reason, or one of the primary reasons that we developed the glutamatergic neurons is that they're an excitable population of cells. And we wanted to have a tool to look at that excitatory pharmacology and excitatory toxicity. Right? And so in order to do that, one of the primary methodologies we use is the MEA. And what we're looking at here is a raster plot of MEA data. Typically, what we use are the 48 well plates from Anson, and what we're looking at in here is one well of activity. All right? So this is a single well of activity. Each row represents a single electrode within that well. Each tick mark of those wells represents the recording of electrical activity in the neurons that are directly lying on top of that electrode. These peaks up here then represent periods of synchronicity where all of the electrodes in the well are firing. That is to say, the message is being propagated all across that well in a synchronous fashion. And this synchrony is really what's thought to represent a well-coordinated electrical activity output that then can be interrogated with a variety of compounds. Right? Now, the real value in having such an interrogated network is that you can look at intracellular activity as well as intercellular activity. So, for example, you can look at and interrogate the ion channel activity. So we're under baseline conditions. We see the firing and synchrony. In this case, you interrupt that with 4-AP or a potassium channel blocker, and you see that level of firing drastically decrease, as you would have thought. Similarly, you can do the pharmacological intervention of sodium channels, calcium channels, really whatever you care to look at. Secondly, then, is looking at that cell-to-cell communication, where again, we've got that baseline activity on a different scale than above, but you can see the periods of synchronicity, and then that can be interrupted or modulated in a variety of ways by chemical addition. Here's the example is PTZ, and we see a really slowing down of that that synchronous activity. All right, so you're really, again, able to do a holistic interrogation to look at both intracellular and cell-to-cell communication. Now, one thing for an assay, it's one thing to show the biology, but that assay has to be robust and repeatable. And here we see an example where, a very typical example, where every well that was plated in that 48 well plate shows high levels of activity. So when you plate the cells out, you know you're going to get a full plate of data, not just a well or two or three here and there. Okay. Now the real purpose of this, is to really then interrogate drugs. And there's a large variety of endpoints that one can pull out. What I'm showing here are really single endpoints. Oh, sorry, let me back up. What we see on the top is really in a dose-dependent fashion, we can see qualitative changes in the way the cells are firing and communicating with each other. Now, we can pull out a lot of those endpoints, such as spike burst intensity, so amplitude. We can look at interspike interval or frequency. We can look at degrees of synchronicity, the level of network synchrony, many individual types of endpoints, some of which are shown down here on the lower right-hand side, really spread out along the x-axis as a series of individual endpoints, and along the y-axis in concentration. So we can see, for example, network velocity tends to increase with the addition of PTX. Similarly, we can look at the types of graphs that compare two different endpoints to see which variables, which endpoints are correlating or tracking with the other endpoints. So again, there's a wide variety of ways you can analyze this and endpoints one can pull out, but then it's really up to the investigator to determine which endpoints are pertinent and specific to their needs. What we'll see with Dr. Bader's presentation is really how you can look at those endpoints as a gestalt, or as the whole, and really do fingerprinting and classify your unknowns into distinct chemical classes and compound classes. So really taking this first level analysis to a higher order. Okay, so I just like to pull the camera back a little bit and look at the big picture, and well, actually not quite yet. We'll have this slide first, a summary slide. We've shown you a couple of examples of chemicals, and what we see here is that these aren't just isolated incidents. At CDI, we've looked across a fairly wide variety of of compounds, whether they're control compounds, excitatory compounds, compounds used in the clinic, as well as other chemically active compounds as well. And again, we'll see how this type of a data set can be expanded into a much larger universe with Dr. Bader's presentation. So in summary, you see the GlutaNeurons are an excitatory neuronal population. They're a seizurogenic model. We've tested across many chemical classes, and are currently under evaluation within the HESI translational biomarkers of neurotoxicity. So really what I've shown you is just a couple of assays that can be utilized with the cells. There's obviously a wide variety of additional types of assays that you want to look at, and in the realm of toxicity, where we, CDI, are able to really provide a variety of endpoints, measurement techniques across multiple cell types and multiple support venues. All right, and pulling it out even further to take advantage of that IPS technology and the power of that, we're able to look at these types of techniques and endpoints across neurodegenerative diseases, a variety of disorders, and a variety of other types of neuropathies. All right, so with that, I would like to say thank you. And at this point, I'll turn it over to Dr. Benjamin Bader of Neuroproof.
Perfect. Thank you, Blake, or Dr. Anson, for this very nice introduction on the iPS models, which we at Neuroproof use for, in part, neurotoxicity detection. As Neuroproof, we come from more than 20 years of background with rodent mouse cultures, and now since about 5 years collaborating with CDI, we have emerged into the iPS field as well. So I will concentrate on the microelectrode array technology, which Dr. Anson was introducing already, but from a point of view that we use the mouse model as a reference to characterize novel human iPS models for understanding maturity of those new models. And that is our expertise here at Neuroproof, is this multivariate data analysis and artificial intelligence-based analysis. Afterwards, I will focus on the functional evaluation of human iPS neurons on the example of CDI iCell products. This is again a topic of maturation and assessing time points for screening assays. The physiological relevance, compound effect, comparison between mouse and human cells is also needed for this functional evaluation. And finally, with this seizure pharmacology assay, to compare seizure prediction in mouse and human neurons. So it's a twofold presentation today, coming from the mouse as the reference model to the advantages of novel human iPS cells. Now, some words on Neuroproof. We are CRO, contract research organization, concentrating on CNS-related projects, and doing this as a service for neuropharmacology and pharma companies on nutrition. More than 20 years with this MEA technology is what we work on, since 5 years in iPS neurons. We have extensive quality standards, since we have high culture application and data analysis here. This includes long-term cultures, long-term recordings, which we combine with our USP, which is a proprietary data analysis platform enhanced by artificial intelligence methods. This is what Dr. Anson was referring to: classification of compounds, database comparison, or side effect prediction. And that is for the seizure realm, the focus of today's talk. But certainly we have several assays established on disease modeling, such as Parkinson's, Alzheimer's, rare diseases, epilepsy, even botulinum toxin screenings and others. And since the iPS screening is also part of huge consortia, we are also associated with IMI, iPSC, and Standby. Now, in the drug discovery pipeline, we have been working on, in all the different stages so far, concentrating on lead optimization and preclinical testing. So our typical workflow is that we have cultured neuronal cells on MEAs. You see this MEA here with the tips of the electrodes where the axons lie on top. They are spontaneously active, so no external stimulation needed. And this is where we record based on the waveforms which were separating the different neurons from each other. Now, eventually, we add compounds to that and profile them in a phenotypic fingerprinting manner and can compare compound profiles with each other, which then are used in our artificial intelligence methods to compare similarities or differences. Now, the readout is, as was already told by Dr. Anson, it's an extracellular action potential on a single neuron level or a network level. And we derive out of this data, besides the number of action potential or bursts, also the number, like more than 200 parameters to produce this fingerprint. And then artificial intelligence-based methods, including cluster algorithm, machine learning, and Z-factor-based parameter integration. This data analysis was already published several times in the last couple of years, which you can have a look on separately, so I will not go into that classification detail right now. Now, using iPS neurons, which come cryopreserved out of a vial, which we just put on MEAs, as you see here, we also use the Maestro recording station from Axiom Biosystems, multi-well MEAs. We record the raw voltage, transfer that into single spike raster plots, and eventually we can see compound effects on a network level. Now, the activity is clustered in bursts, and these bursts have a shape. And this burst shape is described in the top right corner here. So there is a burst plateau, a burst amplitude, an area, interspike intervals, etc. So there are many parameters which describe this burst structure, which are very predictive and descriptive for compound effects. Far more advanced than just calculating or recording spike rate. Besides this, as we have a network, we have the synchronicity and connectivity measure, which is in synchronicity index variation over the network. And besides that, we also have regulatory or oscillatory behavior parameters. So all these parameters allow us to derive a fingerprint of a compound or a cellular model. And this is to just show you how this looks in live mode. Here you have a heat map on top where every electrode which is highlighting is active. You see that they are synchronized, and in the bottom left you see that this activity runs through the analysis window here. So you're seeing that many of those electrodes are synchronously active, and this synchronicity is a measure which we need for addressing maturation. Now, as you see in this bottom right corner is a spontaneous activity pattern, and this pattern is very specific. Here you see that for frontal cortex, it's a 60-second window at 28 days in vitro. But that is different between different neuronal cell cultures, as you see in this slide. So cortex is in the top left corner here, but we also have tissue cultures here from hippocampus, spinal cord, and midbrain, which are very unique. And here you see just examples, but if we use hundreds of data sets and put them into cross-correlation, you see that they are very nicely linking and recognizing each other. So it's a very reproducible spontaneous tissue-specific activity pattern. And we use this database now for actually analyzing unknown activity patterns, such as from iPS neurons. Over the years, we have collected several mouse cultures as reference, which you see here. So these are well characterized by many groups, and there's lots of literature characterizing those mouse or rodent cultures. But we also have screened several of different human iPS culture models, including GABAergic, DOPAMINERGIC, GlutaMatergic neurons, but also motor neurons or peripheral neurons. And we have also screened different genetically associated disease models, such as Alzheimer's or Parkinson's or rare diseases, with this technology. So it's actually a way this technology can be used is to follow the maturation. And if we want to talk about maturation, the functionality readout is a very interesting way. So here you have one example of mouse cortex cells. So they increase the activity over time in vitro, which you see here, but you can also modulate that with a growth factor called BDNF. So it's a synaptogenic marker, which is accelerating that development. So we can use this readout to address how we can improve iPS-derived neurons, but also, and here I have an example showing that we can, or how sensitive this technology can be. In collaboration with an infant formula producer in US, we developed a new combination of infant formulas where we actually added different mixtures of compounds and see this promotion of neuronal activity. Interestingly, this in vitro assessment showed that we had about 20% faster maturation with this compound mixture, which was then also introduced in vivo in a clinical trial. And this showed also the benefit of this mixture because we have this cognition enhancement in this infants. This is a nice correlation, in our opinion, to show the link between in vitro and in vivo results. Now, from a compound screening approach, we use concentration response curves, which we then, you see the spike rate, transfer into this whole complete phenotype, which you see on the seed map. So this is very compound specific, and this is what we then use for comparing compound profiles with each other. And we have more than 400 compound profiles stored in different tissues, tissue cultures. We have assessed the compound combination, used that for repurposing or repositioning and toxicity tests. Now, this is going into this way that we use similarity matrices and pattern recognition tools to actually show similarities of compounds. And in this phylogenetic tree here, you see that the compounds cluster according to their molecular mode of action very nicely. And let me come back to that when we talk about seizure prediction, because that is the technology we use then in both human and mouse cells. So how does it work with human iPS neurons in terms of maturation and relevance from a physiological endpoint? Now, the glutamatergic neurons Dr. Anson was referring to earlier, we've also screened those, and we see the functional development. And in the first place, they are very active already after a couple of days. But eventually, at 14 days to 21 days, they form the synchronicity, which is really needed to assess functional maturity. If the cell network is not communicating with each other, it is not mature enough. So here we use this technology to assess the functional maturity. And on the yield perspective, we have a very high yield on active electrodes over the time of action and recording within the 3 weeks in vitro. And if we, and this is what we do here, Neuroproof, we split up to 2 different neurons per electrode and end up with up to 30 different neurons we record from. So now this pattern here has a certain pattern, and we want to understand how they are physiologically explainable. And in this context, we use our database of mouse rodent cultures, of different tissue-specific cultures, and ask the question, how do compare, or these human iPS neurons compare to the reference models? Now we use this database, classified them, and see a very nice correlation, and use this classifier to analyze novel models like the iCell products. We use that for protecting high quality standards, and this is also needed now to protect the quality and to assess the quality of the iPS models. And eventually, when we ask this question using this classification approach, we see that iCell GlutaNeurons, for instance, have a very high similarity to primary mouse hippocampus cultures. And iCell GABANeurons have a very high similarity to ventral midbrain cortex co-cultures. So that is also the physiological relevance I would need to see if I work with human midbrain-like neurons. And this is also true for other cell lines. And here we show that those iCell products have a very clear differentiated activity pattern, which we can use as a reader for quality control. And if we think about disease modeling, we can even go for modeling Parkinson's in a dish, where we add Parkinson's-inducing compounds, decrease the similarity to this midbrain-like phenotype, And then eventually, if we test a rescuing agent, we rescue that phenotype back to midbrain activity. So this is how we can use this approach of using a well-known reference model to assess the physiological relevance of disease models and human iPS models. Now, how does it come from the pharmacological response? So if we know that iCell GlutaNeurons have a more than 90% functional similarity to mouse hippocampus pattern, do they also compare pharmacologically? And here we have two examples of compounds. It's methoquine and NMDA. Both concentration response curves, what you see here, and 5 example parameters per compound show that cortex in green and isoglutamatergic response in red are somehow different. If I add the response of hippocampus, for instance, I see that those isoglutamatergic responses are more likely and more similar to mouse hippocampus concentration response. So it's a matter of what you compare with each other. So here I show a physiological similarity to hippocampus activity patterns and also a functional response pharmacologically to hippocampus. And this is also relevant for seizure prediction, as the current seizure prediction assays are also used in mouse hippocampal slices. So we have used also this kind of screening approach. Besides what we have tested in cortex already, we have also a selection of seizure respective compounds tested in iCell GlutaNeurons. We have also tested the botulinum toxin, which is a measure for synaptic connectivity, because it destroys synaptic connections. And what we see here is that if we add it after 28 days in vitro, in pico to nanomolar ranges, which is very high concentrated, of course, but here what we see is that the synchronicity actually is abolished by botulinum toxin, and this in a concentration and time-dependent manner. And we see a very high readout in synchronicity describing parameters, even at the 10 picomolar range. So showing that it's a very sensitive readout to investigate synaptic and polysynaptic connections which are present in these iCell GlutaNeurons. Coming from this characterization now to the application on seizure prediction, I start with the problem, and that is seizure liability of standard of care drugs at the market, at the body. And there's this paper from Kumlian and Lundbeck who have described the seizure liability and adverse drug reactions. And you see this list of compounds and drugs and their seizure liability in men. So we certainly want to avoid this with novel drugs, and therefore we need in vitro screening assays, and at the moment one assay is the hippocampal slice assay, and we want to replace that also in the real 3R, replacing the refinement reduction of animal use. Now, if we test a seizure-genic compound, which Dr. Anson was also referring to, it's picrotoxin, it's a GABA receptor blocker, we see the increase of synchronicity and regularity, and we even see these ictal-like events. So there's a hyper-excited activity and increased regularity and synchronicity, which is needed to predict seizure. What you see here is a selection of compounds we used to calibrate the system for predicting seizures in vitro. The top row is a selection of known seizure-inducing agents. They are all excitatory. You see the spike rate as a readout, and the bottom part is 3 compounds which are also excitatory but not seizurogenic. So we want to compare the phenotypic differences between these 2 groups of compounds to actually identify a measure which is very predictive for seizures. Now, if we go just from the excitatory spike rate readout, all those compounds are excitatory, but they are very different in terms of their phenotypic profiles in several birth structure, oscillatory, or synchronicity methods. Now we use this phenotypic difference, which are very distinct in several of those parameters and perform a classification using this artificial intelligence methods I was referring to before. And what I can show here is that those two groups, they separate very nicely, and this separation can be then used to investigate unknown compounds. Those unknown compounds, for instance, are test compounds like oxytryptamine, neuropeptide, or SCH, tool compounds. These ones are all excitatory, but they are safe because they were classified in a concentration response way into the non-seizure class. So if you have a seizure-genic compound, it should end up with higher percentages at the right-hand side. And this is actually the case for tool compounds which induce seizures. And here you see that AMPA or glycine or also acetylcholine classify into the seizure-genic risk class. So this is a tool compound validation. How does it do with marketed drugs? And this is what you see here. The therapeutically relevant concentration range is highlighted in green, and the predicted seizure risk in our assay is highlighted in red. You also see the seizure risk from literature in terms of these asterisks here, or here in terms of percent values from the Comian paper. What you hopefully see is that there is an overlap of concentration ranges used in the clinic and found in patients' brains, and the seizure risk predicted here in vitro. So we correctly classify those compounds, and eventually, if we correlate that with between our in vitro prediction and the percent of seizure risk in men, we see an interesting correlation that, for instance, clozapine has an 88% predicted in vitro with 9% in men, and it's going down to haloperidol with 50% prediction and to 3%. So that's the beginning, and donepezil has sort of lack of seizure prediction, although there are seizures predicted for cases in men. But we should not forget that donepezil is also inducing seizures. That seizures are also symptoms in dementia, so it's difficult to split that apart. So we are able to use this seizure liability prediction in mouse cells. Now, how does it do with the human cells? And we did the same assay with the iCell GlutaNeurons from CDI, used the same panel of compounds and see the same concentration response profiles between those compounds. So all are excitatory, and we even see the same prediction matrix here between mouse and human. So they perform as good as the mouse model in terms of seizure prediction calibration. And if we now investigate a selection of unknown compounds again, what we see is that they also predict the seizure genicity for acetylcholine, naphthalene, or naloxone. And if we compare that between mouse and human iCell GlutaNeurons, they actually can perform better and predict more seizure risk or equally than mouse cells. So this is where we need further investigation, how the differences are. But what we can say is that those cells are very nicely applicable for the prediction of seizure in vitro, and that comparing that with a reference model is very advantageous. Now, what I've shown you today is that the MEA screening combined with multivariate analysis allows the phenotypic screening of different neuronal cultures. It's a system for screening compounds in drug discovery pipelines, and it allows the quantifying of maturity on a functional basis. And this is very important to address the quality of human iPS neurons, especially that those cells which we have in hand from CDI show a very mature functional activity with synchronized activity patterns, which can be used for screening, have a really high yield in terms of high throughput screening applications. And they show this biological relevance, like that a dopaminergic culture is most similar to ventral midbrain of mouse, or to hippocampus in terms of GlutaNeurons. We need the phenotypic comparison between non-seizure and seizurogenic compounds to predict most effectively those seizure liabilities in vitro. This machine learning we have applied here, and the classification allows this prediction in mouse and in human neurons. And by doing that comparison, we see actually the value of the human cells, because we not only use them to replace and refine them, but they might be even more predictive than the current mouse models, which needs to be elucidated. With this, I want to thank you, and we are certainly open for questions.
All right, thank you, Ben. Excuse me. So the questions now, for those of you that have asked questions or typed them in, we have the iCell GlutaNeuron product manager, Dr. Kevin Olstrom, at a different site, who will read off some of the questions for us. So, Kevin, are you there?
Yes, I am, Blake. Thank you for the presentation from both you and then from Ben. That was very informative. So we have several questions, but there are some that if those still that are listening want to still submit them, we will definitely ask that to Blake and Ben. But so for one of the first questions that we had, and Blake, you kind of touched on this with the single cell gene expression of the GlutaNeurons, but the question was, how does CDI assess the quality of their iPSC derived neurons?
Okay, thank you. We do that in a variety of ways. One is through marker expression to confirm the identity. We also have viability metrics to ensure that the culture after thaw is viable and we just don't have a bunch of half-alive cells or half-dead cells. And then last, but definitely not least, we qualify each batch through that single cell marker analysis, as well as through the functionality and the formation of the synchronous activity on the MEA. So each batch is QC'd in terms of viability, identity, and functionality.
Okay, all right, thank you, Blake. Another question just regarding the culture and the timing of the activity development within the GlutaNeurons. The question is, how long should the cells be in culture before they start to fire action potentials, and how long before they are synchronized?
Maybe that's a question which I can answer. So we have screened the iCell GlutaNeurons between 3 and 35 days. And to address this assay time point, we need a synchronization. So after 3 to 5 days, they are already active at several electrodes, but they are not synchronized. And that synchronization starts at 14 days. And we usually use those cells between 18 and 21 days, because there we have them synchronized robustly at all wells. But they can certainly be used also at 35 days and longer.
Okay, thanks, Ben. And then another question, and this is pertaining specifically to a part of the CDI presentation, but then I'm sure, Ben, you can speak on this as well. But the term network velocity, could either of you just elaborate on the meaning, or how does one use that value to interpret data?
Kevin, you have stumped me. So network velocity comes out of one endpoint from the suite of endpoints that Axon provides in their neurometric tool. Again, I believe it is a measurement of how robust that network activity is. So whether or not you've got a few electrodes in synchrony, or whether you've got a strong, high level of synchrony across the well.
All right, thank you, Blake. So then a question regarding excitotoxicity. Has CDI or has Neuroproof looked at excitotoxicity in the context of leading to neurodegeneration? So said another way, has CDI looked at the excitotoxic response of various compounds?
Maybe I can answer that from our Neuroproof perspective. So we usually screen compounds at a very high molar range to see when the compound actually kill off neuronal activity. So we know that the loss of active electrodes is linked to cellular health. Maybe if you correlate MTT cytotoxicity assays with the number of electrodes, that goes down. So high glutamatergic agonists at higher concentration, they tend to decrease the number of active electrodes and therefore also the cellular health. So that's what we can say, that this is then a readout where we can use the number of electrodes as a measure to look into excitotoxicity, and the activity drops down also. I had one of the slides here. Blake, you want to add?
Yeah, no, similarly, we've done some work on excitotoxicity within CDI as well as other customer sites. And there was a poster presented at the 2017 Society of Toxicology meeting by one of our customers that delves into that. So we do see the cells being used as a model for excitotoxicity as well as seizurogenesis. And feel free to drop us an email and we can follow up in detail on that.
Okay, thank you. So on oh, sorry, was there a question there?
No, just clearing my throat, sorry.
Okay. All right. Yeah, so another question then, has the GlutaNeurons and just the general MEA approach to looking at electrical activity been used to investigate synaptic plasticity? And if it has, are the mechanisms similar to brain slices?
Well, that's a real cutting edge research question. I'm going to toss it over. Ben, do you have an answer?
Yes and no. So,
okay.
So the technology in general can be used, and there the Axion system is actually quite nice to use because we can stimulate electrodes electrically. So, and we have used that with at least primary neurons with lots of interesting results on a plasticity panel, so that we can see an increase of responsiveness to stimulation by compounds, for instance. So that is not the same what you usually do in slices if you talk about LTP or connectivity measures, but it is certainly possible to use this technology in combination with in vitro cell cultures to address plasticity in an indirect way.
Okay, yeah, thank you, Ben. Yeah, to add on from my end, that's something that we're interested in investigating. And so, as Ben said, this is something that the Axion system enables one to interrogate. And so, this is just an exciting new area to be looking into. And so, for Ben, you touched on it, Blake, you touched on it as well. I think a thing that people are always interested in understanding is the comparison between primary neurons and then iPSC-derived neurons. And just, do you have anything you would want to elaborate on in terms of comparing their functionality, or how you see the two being used in the future?
So, in our opinion here, the reference is needed to really judge on the quality of the neurons, of the human neurons. So if I do a compound screen, for instance, just in human cells, maybe let me tell that as an example, I see maybe a 70% effect size in the human neurons. So how can I say this is good? Maybe, why is it not 100%? Now, if I use a reference model like mouse, and there I see maybe just a 30% effect size, then the human model all of a sudden is much better than the mouse model. So this is how we can use this reference in mouse model. And I would say from our customer base, we see that more and more people jump on the human iPS neurons, even as standalone, but several are doing that side by side with rodent and mouse and human models. So I think it will not be replaced completely, but can be used as a good reference model and to judge on the quality of the novel human iPS neurons.
Yeah, if I can add to that, as Ben's presentation really highlighted, there's a strong database of the mouse work to which the human cells as a new model is being compared to. And we already see areas where this new model is surpassing the benchmarks of the mouse model. And then on top of that, one of the small little panels that Ben also presented was looking at that Parkinson effect. What we can do, or others can do, with the human models is now really incorporate that clinical background or the patient background to investigate these modes of toxicity and therapy and biology. So obviously we wouldn't be giving this webinar if we didn't believe in the human model. And I see from here it's only going to grow and be incorporated into more and more avenues and generate more and more broad bases of data.
Yeah, thank you. So there are two other questions that came in regarding the expression levels of histamine and serotonin receptors, as well as the mean firing rate of the GlutaNeurons at 21 days in culture. And instead of asking these two to answer that question right now, what I will do is I will follow up with both of you who asked that question to give you the specific information for that, because we can provide gene expression for the two receptors in question, and then also I can provide the information on the mean firing rates of the GlutaNeurons. But in terms of additional questions right now, let me make sure that I've covered everything right here. If anybody does have other questions to ask, please submit those. If you don't have any right now, as Blake and Ben mentioned, you can send questions to us at CDI at the support@cellulardynamics.com address, as well as then at Ben's address at Neuroproof. And so I think for questions, we don't have any additional right now, but if you have others, please feel free to send our way, and we will get back to you with the right information then.
Thanks, Kevin.
Yep.
Yep, thanks, Kevin. And so I think in closing, I'd just like to mention that this webinar will be up on our respective websites, and when that happens, we'll send out a group email to all the registrants to let them know when that's there. And so with that, I'd like to thank Dr. Bader and Neuroproof for hosting us here and for participating in the joint webinar. So thank you very much.
Thank you. To CDI also. Thank you.
Thank you, everybody, for joining us. I wish you all a good week and a good December. Thank you much. Bye-bye.
Bye-bye.